This is a strip of Panadol, right? Oh, thanks. And if you have a headache, you just pop a couple of these into your mouth, and half an hour, 1 hour later, the headache is gone. So it's magical. So what is the magic in the pills? And how do you know it's safe for you? And how do you know how many pills you should take for each time? So I will try to answer all these questions in this next half an hour. Yep, my name is Chung. I work in a center in A-STAR by the name Experimental Therapeutics Center. So we experiment with new drugs and see which one can be a new, will potentially be used in human in the future. So before I begin, I will tell you a story first. Alexander Fleming, he discovered that some fungus can produce something called antibiotics to kill bacteria. That was in 1928, right? So he wanted to produce more of this fungus juice that he called at that time so that he can test it in human. But for 10 years, he couldn't produce enough fungus, and he doesn't have the skill to purify the fungus juice from the fungus to test it in human. So for 10 years, he couldn't do that. So after 10 years, 1939, that's the time, the beginning of World War II in Europe. So a bunch of scientists and chemists and clinicians come together in Oxford. They offered to grow up the fungus to produce the drug. So they can produce up to 500 liters a week. And they hire people that are called penicillin girls to produce all this. So at that time, they managed to produce enough of this fungus juice, which we call antibiotics nowadays, and they test it in the patient. So they saved a lot of lives of the soldier during World War II. So that was like almost 80 years ago. So fast forward to today, is 80 years from later. The medicine are more complicated. What we understand about biology is more complicated. So we need more manpower and more team player to come together from different people from different fields to come together so that we can produce a drug. So you will see that. So the take-home message for this slide is there is no Spider-Man, Batman, Iron Man in this field. In this field, you need a team. So you need something like a transformer, a group of robots, human. And there are a few outstanding characters like Optimus Prime and Bumblebee, but it's a team effort these days to discover new drug. So this is a workflow of drug discovery, from the concept, from the disease that we want to study, right up until clinical trial in human. And these are the groups of scientists that are involved. So each dot here represents a different group of scientists with a different expertise. So you can see that you need many different scientists with them, many different expertise to come together to finally come up with a drug. I will go through this one by one in the subsequent slides. First, target. The disease, your cell is like a bag of soup. Inside there are many ingredients, many proteins. A disease is usually caused by a protein that has gone haywire, and it causes a disease. So we call that protein that goes haywire a target. And we want to target, we want to hit the target so that we can stop its function. Okay? So a medicine is like the arrow. You will hit the target, hit the protein, and stop its function, and that is how you cure a disease. And more terminology. Medicines, there are many ways to treat a disease. For example, you can use antibody to treat a disease. In this example here, this is called a Herceptin. This is an antibody against a subtype of breast cancer. Or you can use hormone to treat disease, like insulin, growth hormone, all this. Or you can use cell therapy. Bone marrow transplant is one example. This is a newer example where you can harvest the immune cell from the cancer patient, modify it so that you recognize the cancer and put it back into the patient so that you can kill the cancer cell. So these are ways to treat a disease. What I want to talk about is small molecules. Small molecule, for example, is Panadol. And small molecules are chemical entities, chemical that is not found in your body that we created so that it can block the target, block the protein that we want to stop its function. So I will talk about small molecules in this. These are all different ways to treat a disease, and they are all equally good, just that my expertise is in small molecules, so I will talk about this. And also, small molecule is the most common way to treat a disease. Most of you have taken lots of pills in your life before. So this is still the most common way to treat the disease. So what are small molecules? Glucose is a small molecule. We eat glucose. It comes from your rice. The carbohydrate break down into glucose, and they produce energy. This is glucosamine. The difference is just here. The OH group, hydroxyl group, flip up, and this hydroxyl group become an NH2 group, become an amine group. So the functions are totally different. So this produces energy. This is a protein. This is a molecule that is used to build other proteins in your body. Okay, so they play totally different role. So the difference is just a little bit of changes in their shape. So is our small molecules. So this is Panadol. This is how it looks like. And how does it cure your headache? It basically goes into your brain, block an enzyme in your brain called cyclooxygenase. Cyclooxygenase produces another molecule called prostaglandin that will cause you to have the pain feeling and the fever. So this molecule will block cyclooxygenase, so no prostaglandin will be produced. What is this? What is Lomotil? Any idea? Most of you will have taken it before, or all of you will have taken it before. Yeah, good. When you have lao sai, diarrhea, you take this. So this is different from this, right? So how does this work? This binds to nerve in your gut. So it stops your gut from moving. When you have diarrhea, it's because your gut is moving too fast, so you can't absorb water, so it comes out as watery stool. So this thing blocks the nerve that stimulates your gut, so your gut will not move so fast, so you have more time to absorb water. So different shape has different function. Okay? Okay. So, okay, before I start with this, different shape have different functions. So your protein, you have about 20,000 protein in your body. So one of them go haywire and cause a certain type of disease. So each protein has a different shape. So like a lock and key example. So to open a lock, you need a special key. A key with the right shape to open it. So same with this. If you want to stop a protein's function, we have to have a small molecule of a correct shape that binds to your protein. So this is a lock and key function analogy. And in our center, I have a collection of 500,000 of these small molecules. So 500,000 of these, all with different shapes. And they are stored in plates like this. And because we have 500,000 of them, they are stored in plates that are then stored in shelves like this. In my lab, there is a robot that moves along this aisle to pick up the compound, the small molecule that we want. So each molecule has a barcode to it. So the molecule that we want, we key in the barcode, the robot will move along here to take out the molecule. So the more you have, the higher chance of it, finding one of it that will hit your protein, right? You can understand that? So the more of this different shape, just like if you have a bunch of keys, one of them will be able to open the lock. So same analogy. So the plates are like this. This is a 12 by 8 centimeter plate. And each of these plates has 384 wells. And you can store 384 of these different small molecules. So I will pass the plate around so that you can take a look how small it is. Just spread it around to the back. Yeah. Yep. Doesn't matter. Yeah. Yeah. Yep. Just press it around the back. So we have all these molecules stored in tiny plates. So how do we test them? So let's say we have a protein. We have a protein that we want to block its function. So we want to test each of these individual molecule to that protein to see whether they can block it, right? So, but we have 500,000 of this. How are you going to test it one by one? So how are you going to test it one by one? You can hire a staff and test one by one, but this is going to be very slow. So we have robots that help us to perform the experiment. So with that tiny plate, one plate can perform 384 experiments in one plate. And you can have robots to help you to dispense all the chemicals that you need to set up the assay to measure the activity, right? But because we have hundreds of thousands of these plates each time, so you need a robot to help you to handle them. If not, you have to hire someone to stand beside the robot and move the plate one by one. So we have robotic arms that can do that. Next, you can chain up all these robots into one big workflow. So this is how we do drug. We find out which of these compounds can block your protein. So this is called high-throughput drug screening. Okay? Because there are way too many, so you can't do it one by one. It will be way too slow. And we can do this. My scientists in my lab, one person can test 16,000 compounds a day, 16,000 of these small molecules a day. So, and in about 6 weeks, they will have tested 500,000 compounds. So without the help of these robots, you can't do that. So this is how the robot works. Yep. So this is the place where the experiment is being assembled. So the robots have to move the assembled plate to another place so that they can put in new plates. So the movement is a bit slow because all the plates will have liquid in them. So if you move too fast, you may spill them. So the movement are deliberately slow. So you cannot compare this to a wafer fab manufacturing factory. Yes, so that this robot actually assemble all this, and then the other robot at that side, they are actually adding in all this, your enzyme, your protein, your molecules, small molecules, and then the detection reagent, everything on that end. Yeah. Like this robot, it can handle 96 wells within that at one go. And there are robots that can handle all 364 wells at one go. And this day, we are actually going into 1,536 wells per that small plate. So that at one shot, at that small footprint, you can perform 1,500 experiments. So the experiment has been assembled at that site. Then you come over to this site where the plates are sealed so that you can go into storage or incubation for subsequent work. So this is how it looks like in one part of our workflow. So this is how you assemble such a small, so many experiments in such a small footprint. So you need the help of robots. So first, I will repeat that again. So first, we know what protein we want to target. So that is let's say in this example is a cancer target. Then some scientists who are specializing in development assay will develop a miniaturized experiment into that small footprint. Then they send to us in the high-throughput screening group, where we use the robot to assemble and screen the molecule in a very high turnover manner. Then after the screen, you will have molecules that will block your protein. But this is not your drug yet. After the screen, you will definitely, of the 500,000 compounds, definitely some of them will bind to your protein and stop its function. But that is not the final drug yet. We still have to improve it. At this stage, we do not know how efficient it is in the human and whether it has side effect or toxic effect to human. So this is just the beginning. You found something that will work, will block this protein here. So you go into this step where we call heat optimization, where we optimize the compound so that it is more efficient, less toxic to human. So for this step, you have another group of scientists that come into play. So this step is called heat optimization or heat to lead phase, where you have chemists, medicinal chemists, scientists that specialize in nuclear magnetic resonance or X-ray crystallography, people who test the effect in animal, called preclinical pharmacologists, and biochemists who study the physical effect of the chemical reaction in your body. So NMR scientists and X-ray scientists, these are scientists that study the molecular interaction. That means they study at the atom and electron level how the molecule interact with your protein and how it stops the function. So this all requires specialized knowledge. So you see different groups of scientists that are different from this group of scientists will come in and make this drug to be more effective. So I will take example from the medicinal chemists and clinical pharmacologists in the subsequent slides. So how do we make the drug better? We do something called, in the heat to lead phase, we do something called structure-activity relationship, SAR. So this is a real-life example. This is a drug by Boehringer Ingelheim, a German drug company. This drug is used for treating heart failure. In heart failure, your heart pumps very weakly. It lost the strength to pump and relax, so you cannot circulate blood throughout your body. So this is called heart failure. And heart failure can be due to many reasons. But one of the protein in your body that's responsible for the weak pumping is called RSK2 protein. Okay, so they come up with a compound, a drug that can block this RSK2 function. But it has a side effect. The side effect is it also blocks another protein known as alpha-1 adrenergic receptor. This protein is responsible for your blood vessel strength. So by blocking this, your blood vessel relax. So you have hypotension, low blood pressure. So this is an unwanted side effect. So the chemists, they will do something called structure-activity relationship to make this better. So they will break up this protein into the left side and the right side, and they modify a little bit each time. See, this is at first like this, and they added something here. And this is, at first there is no nitrogen here, so they added nitrogen in. So at each of these steps, they will send the protein, they will send this compound to a group, another group of scientists to test its function. So they change something, they test again. They change something, they test again. So this is the right-hand side. What do they do? What's the difference? Anyway, yeah. They remove this group and they add a lollipop here. This is not a lollipop. This is called a phenyl group, but it looks like a lollipop. So anyway, the chemists, they will modify a little bit and we test it again. So until finally, in this example, after 15 tries, they come up with this shape, which is slightly different from this shape. But at this time, it's more potent. You see here, it can block this enzyme RSK2 at 1.1 nanomolar. But now after 15 tries, now it can block this using just 0.2 nanomolar. So the lower concentration that you use means to produce the same effect means it's more potent. And at this stage, it doesn't interfere with this alpha-adrenergic receptor anymore. So it won't cause you to have low blood pressure. So this is how you make the molecule better. This is an iterative step. They change something, we test it again. They change something, we test it again. And this example, there's only 15 tries, and they managed to make it work. In actual fact, normally we take 100 to 200 tries to get something to work, or you just never work. So this is one example how the chemists make it work better. Okay, the dose makes the poison. a wise man in the 16th century said that everything is poison and nothing is without poison. It's the dose that make the difference, right? So for example, snake venom. Snake venom is poisonous, you all know that, but you can use it to treat diseases. You can use snake venom to treat, some snake venom can be used to treat cancer, some can be used as antibiotics to kill other microbes, and some can even be used for regulate or prevent a heart attack. So snake venom is good. But if you are bitten by a snake, don't think that you get a free treatment, right? So you better go to the hospital fast, because the dose that the snake gives you is not the dose that the doctor gives you. So it will not work. So the dose is the difference. So how do we know that just now the drug in the previous page that I've shown you, how do we know that what dose we should use in the human? So we send it to a group called preclinical pharmacology group. The pharmacologists will test it in animals first. We won't test it in human first because we do not know how dangerous it is. So we test it in human first, animal first. So we test after the animal ingests the drug, they kick in the drug, where will it go? Whether it will get in through the guts, and after that it get metabolized in your body, so will it remain intact? And whether it will go to the site, let's say the cancer that you want to treat, and how long it will stay in the body before it come out, and what other funny things besides treating the disease that you will do. So we test it in progressively larger animal. So we test it in mouse, rat are larger mouse, and then we test it in a dog, and finally in a primate. But don't worry, most of the time we have enough information at the mouse and at most the dog stage. We hardly ever need to go into primate. So from there, we can extrapolate the effect in the animal to the human. So this part is done by a group of scientists known as the pharmacologists. So after a few years of testing, the chemists change the molecule. We test it again, and these people check the interaction of the atom and electron with the compound to a protein, and these scientists check the effect in the animal. And then finally, after a few years, actually it's more like 4 to 5 years, we will have a drug candidate that's ready to go into human. Then we need another group of scientists. This group of scientists is found in another A-star institute called D3. D3 stands for drug discovery and development. So they are scientists and clinicians that will conduct more toxicity trials and before they go into human. And if the drug is okay, they will help to file the drug with HSA, Health Sciences Authority, or FDA, so that it can be marketed. So you see there are many different expertise involved here. So what I have told you just now is just 1/3 of the journey. So in the lab, we come together to decide what protein to study. We make the compound, we do the screening using the robots, then we test it in mouse. Then if it's safe, it's ready to go into a clinical trial. Phase 1 trial is where we test the safety of the drug. So we just need to recruit about 20 to 50 healthy volunteers and test whether that is safe in them. Then we get a small group of patients, which is usually around 200, 300, and to see whether it has an effect in the patient, if it can cure the patient. If the drug is still okay, it can treat the patient, we will conduct a much larger scale of trial. So this is called phase 3 trial, where we include thousands of patients, and we test for many years to see that whether the drug has a long-term effect, besides the effect that we want, whether they have long-term side effect or this. And if it passed through that, then we will register the drug and it will become a drug in the market. So this is a long road and it takes many, many years. Okay. So how long does it take? The discovery phase is what I told you in the things that happen in the lab just now. It takes about 5 years. Then the phase 1, phase 2, phase 3 clinical trial in human takes about another 10 years. So altogether, it takes about 15 years before you see a drug in the market. And we have to screen, just like I told you just now, we screen like 500,000 compounds in order to get, almost finished, 500,000 compounds in order to get one product. So, but it's not every time we screen 500,000 compounds, we will have a product. It's more like we have to do 20 times of this to get a product. So it's very labor intensive. and it costs a lot of money. To produce a drug, it costs about $1 billion US. So why are we so stringent with our selection criteria? Why do we fail our own drug? Why do we fail our own effort? Because we want to make sure that it has really minimal side effect, it's safe, and it's easy to make. If it's not easy to make, it's going to cost you more money. Whether it should not have adverse effect, and it should be absorbed into your body well, all this. So we make it very stringent on ourselves so that the drug that you see here is really working. So actually, each drug, even including the humble Panadol, is a product of toil, tears, and sweat for a lot of scientists here. Each word here represents a group of scientists that study different things. So you involve many people with many different expertise. When we finally produce a drug, this is what we say. So what does column say? My precious. Yes, it's really precious. Don't underestimate the simple Panadol. Really, really. So what has Singapore achieved in the past few years? Our center, we come up with the first publicly funded drug in 2 years ago in 2015. So this is how the molecule looks like. We take colon cancer from patient, and we graft it onto a mouse. So we put the colon patient from human onto a mouse, and then we test it in the mouse. So with our treatment, the tumor shrink. Without treatment, the tumor will keep growing. So now we are in phase 1 trial in patient. So as long as the drug blocks something called wing production, you will block the cancer that's induced by this wing. So you can use it for cancer like colon, ovary, and pancreas. The second drug from us was just announced earlier this year in May. So this is a drug that looks like this, and it's meant to treat a certain kind of leukemia as long as this signaling pathway in the body. Our cells are made up of many different proteins, like 20,000 proteins, and they form a network of interaction. So something like MRT map. So if you block one path, you can block, just like what happened 2 days ago. The train break down somewhere, so you block the whole flow. So yeah, this is the same. So this drug blocked here. So as long as any cancer that utilize this pathway to cause cancer, we can block it. So our first treatment is leukemia, and we are now looking into extra treatment like diabetes and even autism with this drug, because they utilize the same pathway. So okay, one last advertisement for A-STAR. EDC is right smack in the middle of all these A-STAR institutions, academic, and hospitals. So we make drugs. Then all these A-STAR institutions help us to analyze them. And then we engage HSA, Health Sciences Authority, and what do you call that? A clinical research organization to conduct trials for us. And if it's successful, we will either spin off the drug, we spin off the group of people who invented the drug as a company, or we can sell it to a big pharma company. And this will bring in a lot of money for Singapore. One drug will, before it reaches the market, it will worth a few hundred millions. So this is how A-STAR is using research as an economic pillar. So for the student among us, if you want to be a scientist in the drug discovery field, what you should do? You should get a degree in medicine, pharmaceutical sciences, chemical sciences, biological sciences, computer, law, and business. But if you want to be in the lab, doing the experiment yourself, you should study medicine, pharmacy, pharmaceutical sciences, chemistry, or biology. And if you want to have more control over the project that you do, you want to dictate the direction of a drug, get a PhD. Yeah, and in case you wonder how scientists look like, these are us. I'm here. We come from all over the world, basically all continents, including Africa. We have people, we have scientists from all continents. So we all look normal. Yeah, in case you wonder.